So phospho-ERK. Phospho-ERK is a protein that's associated with survival. When ERK is phosphorylated, you're doing well. When it's not phosphorylated, things are not going well for that cell. And what happens is as soon as you have a death cascade initiated, you're going to have dephosphorylation of ERK, which leads to dephosphorylation of a Krebs protein, which is basically just bad news for whoever's involved. The cell is going to destruct. It's not a matter of, at that point, it's not stoppable. So what we did was we took in a series of antibodies, and we stained for phospho-ERK after I'd taken some dendrites from rat pups and put them in a dish. And it's awful. I don't know, that sounds terrible, but let me just really about it. So at that point, so what you're seeing, the green is a secondary antibody that I added so that I could see where this phospho-ERK is being localized within the neuron. And then I did a series of different tests. Synaptic activation. So this is a sample of a neural cell as it normally would be. So just the synaptic activation area of the neuron is activated. And this is what would represent physiologically as a healthy neural cell that is undergoing synaptic plasticity and is able to form new junctions, and basically what the majority of your neurons are doing right now. Extrasynaptic is what your brain looks like when you're having a stroke, or what your neural cells look like when you're having a stroke. You can see that the phospho-ERK is no longer visible in the axons or the dendrites. It's localized to the somal bodies, and the intensity. The neat thing about immunofluorescence is you can get a rough estimate of concentration based on an immunofluorescence intensity. So you can see not only here is there less of the phospho-ERK in other parts of the neural bodies, because there are dendrites all around here. There's just no phospho-ERK at those sites. The the fluorescence intensity as well is decreased, which means you get a downregulation of phospho-ERK. The very interesting thing about this, though, is that when you treat with Con-G, it looks like synaptically activated cells. So this is a person who is having a stroke. This is a person who's having a stroke who's been given Con-G. This looks remarkably similar to synaptic activity. So this is big news. This is fantastic information, because this means that if you are able to administer Con-G at the right time, you can prevent neural cell death. The snag is that if you give Con-G to healthy cells, they end up looking like extrasynaptic ones. So, which would then prevent synaptic plasticity, i.e. your ability to learn, your ability to form memories, or basically a lot of fun things that most people like to do with their brain. So, it's imperative that you only, if you were going to use this in a clinical environment, you'd have to be very specific about when you're treating the patient with this compound. Because it does fantastic things, and studies have shown in mice that it can prevent neural damage, but you have to be really careful about what you're doing and when you're doing it. So I think on my next slide I have a graph of immunofluorescence. So this is basically the computer that I was using was able to measure fluorescence intensity and of phospho-ERK, so this survival protein basically. The more phospho-ERK that you have, the better you're doing. Synaptic, healthy neuron as it should be. Synaptic plus G imitating extrasynaptic activity, i.e. when you're having a stroke. You can see that those are very similar. There's a larger amount of phosphorylated ERK, so better viability of those neurons when you treat with Con-G in extrasynaptically activated cells than even in the wild-type synaptic. Well, wild-type, but standard form synaptic. So Conantokins G has huge potential to mediate development of Alzheimer's and also prevent neural death after a major stroke or ischemic insult. And that's why it's so cool.
So what exactly is the mechanism of stimulating those extrasynaptic versus the synaptic? You said it was a chemical something?
Yes, it's a kind of evolved protocol. It's by a gentleman named Ivanov.
Yeah.
It's a series of different washing procedures that you would, I think.
I guess I'm asking, how does it work?
How does it work?
Because they're the same receptors, right?
Right.
So then how do you isolate some receptors from other receptors when they're in the same area?
Which is why Ivanov is really, really interesting, because based on an entirely chemical process, he's able to selectively activate certain, it would make no sense. But you also have to remember that physiologically speaking, when you're having a stroke, only your extrasynaptic NMDA receptors are activated. Versus when you're doing well, it's only your synaptic ones. So I don't know how he came across this. Whether he was looking for this or not, but he found a protocol that chemically mimics what you see, that differentiation of what you see physiologically. So how the mechanism works, we have no idea. This was just a brilliant, wonderful theme he fell upon and has been publishing many papers on it since. So look up Ivanov and he'll give you the whole protocol. He's not shy, I found, which is
Daniel? You mentioned that I guess the carboxyglutamic acid is one of the reasons why that it's so expensive.
Yes.
Do you know if anyone's like trying to get like a microorganism to express it recombinant, like the proteins that produce it recombinantly kind of thing?
I don't see why that wouldn't be a good idea. So far as I know, no. Unfortunately, because our lab didn't do absolutely everything ourselves, we got them as cartridges. So I don't really know how they made them. They just came in these cartridges.
Okay.
So I would assume so. I mean, that would be, if someone sat most of us down and said, okay, make GLA, go. I think most of us would look at an E. coli expression system for simplicity and yield. So I would assume that'd be viable.
All right, thank you.